Good evening, everyone. I welcome you all to the live problem solving session on NPTEL course for Bioinformatics Algorithms and Applications. So, today this is the first session for week 1 problem solving. So, myself, I am Ramakrishna Reddy, a PhD scholar from Department of Biotechnology, IT Madras. So, I am currently working under Professor M. Michael Gromia, who is the current course instructor. So, in this course, initial part of the session, I'll provide summary of the previous week lectures, a small discussion. Followed by that, I have prepared some sample problems, so which will be displayed through the slides, and the attendees can answer them using the chat box. Followed by that explanation. Then, if time permits, I'll also discuss about the questions that are posted on Swayam portal. Followed by that, I'll take questions from live audience. So, any doubt or any clarity, students can use the option of chat box. Okay, to start with, concepts and importance of bioinformatics. So, what is bioinformatics? In field of biology, we define bioinformatics as the combination of biology and technology, where computers become our trusted allies in unraveling the mysteries of living organisms. Imagine a world where complex details of biological data are seamlessly organized and managed with the help of advanced computational techniques and information technology. Well, that's precisely what bioinformatics is all about. So, what areas of knowledge that bioinformatics include? Bioinformatics is highly interdisciplinary field that draws upon various domains including biology, medicine, mathematics, physics and computer science. So, here the role of biology, the biology acts as a foundational science in bioinformatics, providing the raw data and context for understanding living organisms at molecular, cellular and organism levels. Biological knowledge is important for development of computational methods and algorithms for the analysis and interpretation of biological data. Next, medicine plays a crucial role in bioinformatics by integrating genetic and molecular information into the clinical research and practice. Bioinformatics in medicine involves applications such as personalized medicine, pharmacogenomics, and the analysis of disease-related genetic variations. The other one is mathematics, which provides the theoretical foundation for many bioinformatics algorithms and statistical methods used in analysis of the biological data. Mathematical models are employed for tasks such as sequence alignment, structure prediction, statistical analysis of large-scale genomic data. Then we have physics, which contributes to the understanding of structural aspects of biological molecules, particularly in structural bioinformatics. Finally, we have the core subject, that is computer science, which provides the tools and techniques for data storage, retrieval, analysis and visualization. Algorithms and software applications are developed to process and interpret biological data efficiently, ranging from sequence analysis to systems biology simulations. So, if I want to summarize the interdisciplinary nature, so bioinformatics acts as a nexus leveraging the insights from biology, harnessing the computational power of computer science, utilizing the mathematical models and incorporating principles from physics, medicine to advance our understanding. So, these are the various subjects that comes under the interdisciplinary nature of bioinformatics. So, next, to start with, major aspects of bioinformatics that contribute to the advancement of biological research and understanding are well organized databases, computational derived hypotheses, web servers, virtual screening and big data analysis. So, here bioinformatics aims to create and maintain comprehensive, well organized databases containing biological information such as DNA sequences, protein structures, and gene expression data. So, what is a database? Can anyone answer through chat, or are they free to unmute themselves to talk?
Like collection of information in a sequence.
Yes, yeah, thanks for the answer. Can you give any database that we use in our day to day life?
Google.
GenBank.
Yeah, okay, GenBank. So, for example, our mobile is a database which stores the information, list of our contacts, list of organized way of our gallery, albums. So, this is one regular example of a database.
Google.
Yeah, Google, Unicode, yeah. Yeah, okay, thanks for your answers. So, what these databases do? So, these databases facilitate efficient data retrieval and analysis, enabling researchers to explore and understand various aspects of biological systems. Next is computationally derived hypothesis. Bioinformatics methods helps to identify patterns and relationships and potential correlations within the biological data, leading to hypotheses that can be tested experimentally and contribute to a deeper understanding of biological process. Followed by that, we have web servers. So, under this concept of web servers, so these web servers provide a user-friendly environment or platform that enable researchers to access and utilize computational tools and resources remotely. Or these web servers are sometimes alternative to the experimental studies. For example, we have web servers to predict the stability of a protein, that is to predict the free energy of a protein, or also we have web servers to predict the binding affinity of two interacting proteins or any binding affinity of any protein complexes, and we also have web servers to predict whether a given mutation is a cancer driver mutation or a passenger mutation. So these give us a alternative to experimental studies. Then we have virtual screening. So virtual screening is used to predict the interaction between biological molecules such as proteins and small molecules. So this accelerates drug design by computationally accessing the potential binding of molecules, facilitating the identification of promising candidates for further experimental validation. Followed by that, last one we have is big data analysis. So bioinformatics address the challenges posted by vast amounts of biological data, aiming to develop methods for efficient uh, big data analysis. So now I have a question for you. So which interdisciplinary aspects that bioinformatics leverage to advance our understanding of living organisms? Uh,
Like all of the above.
yeah,
All
people
of
can
the best.
provide answers through chat box. Sorry to interrupt, but kindly provide your answers using the chat box. Yeah, yes, yeah. The answer is all of the above because bioinformatics is an highly interdisciplinary field that draws upon various domains including biology, medicine, mathematics, physics, and computer science. So the answer is all of the above. Next, I have one more. What is the primary role of computer science in bioinformatics? Yeah, yeah, okay. Before moving on to our next question, uh, so in all my lecture starts with the summary to the previous week lectures, followed by some sample questions, then questions from the portal, followed by that at end of my session, and most of my sessions will have a practical explanation of a practical session. So in this session I'll be explaining about the Emboss web server, so how to use Emboss web server for translation, tra- how to find the complementarity and all those for a small sequence analysis study. Yeah, so the role of computer science in bioinformatics is providing tools and techniques for data storage, retrieval, and analysis. Then I have one more. What is the objective of virtual screening in bioinformatics?
Excuse me, sir. What is the meaning of virtual screening?
Uh, yes. So, which virtual screening is actually mostly employed in drug discovery. So, we will use it's a potential way to predict the binding of a molecules, like n number of molecules to a like small molecules to a biological molecules, like how these proteins, for example proteins and small molecules bind. So at a time you screen multiple.
Okay, sir. Sir, you mean the receptor ligand interaction?
Yeah, so receptor is a protein, ligand is a small molecule. Yeah, in my
Okay,
explanation.
sir. Thank you, sir.
Excuse me, sir.
Yes, please. Yeah.
Sir, I have a question related to the first question.
Yes.
Sir, in first question there is a option that is mathematics and physics. So I do want to know that how this bioinformatics is related to the mathematics and physics.
Yeah, so here mathematics, generally mathematics we have statistical methods or algorithms that are used to develop or used to assess the performance of the biological models. Followed by that physics, the concepts of physics such as Newton's laws are widely used in simulation studies of the protein.
Okay, sir.
Sir, what is the name of the law which you just told?
Yeah, we use Newton's laws in simulation of studies, sorry, GROMAX in protein simulations,
Okay,
like
sir.
F equals to ma and all. So this is the how uh, the in vivo movement or in vivo conformational change of a molecule is studied in simulation studies. Yeah, so virtual screening is used to predict the interaction of a biological molecules with a small molecules. So this biological molecule has a receptor ligand, one receptor and many ligands. So virtual screening simple, but one receptor binding to many ligands. Yeah, so before next is complexities in biological systems. So central dogma is a process in which genetic information flows from DNA to RNA to make final functional product protein. So can anyone tell me what are the steps in central dogma?
DNA to mRNA through transcription.
Okay, after transcription.
mRNA to protein through translation.
So Thank you for the answers. Yeah, so central dogma of molecular biology is a framework that describes the flow of genetic information within the biological system, particularly in cells. It was first proposed by Francis Crick in 1957. So the central dogma outlines three main processes that governs the transfer of genetic information. So it includes replication, transcription and translation. So replication, gen- generally the genetic material is usually in the form of DNA, is duplicated to create an identical copy. This occurs during cell division, ensuring that each daughter cell receives a complete set of genetic information. Followed by that we have transcription. In transcription, a specific segment of DNA or a gene is used as a template to synthesize a complementary RNA molecule. The RNA molecule called mRNA carries the genetic code from DNA to the cellular machinery responsible for protein synthesis. Then we have translation. Translation takes place in cellular structures called ribosomes. During this process, the information encoded in mRNA is read by other RNA molecules such as tRNA, which brings specific amino acids to the ribosome. These amino acids are linked together to form a protein. So translation is the final step in flow of genetic information, resulting in the synthesis of functional proteins. So proteins are the molecular machines that perform wide range of functions in a cell and organisms. So bioinformatics, as I mentioned, is a multidisciplinary field with various subdisciplines that focus on different aspects of biological data analysis, interpretation and application. Some of the prominent subdisciplines within bioinformatics include genomics, which focus on the study of entire set of genes within the organism. Then we have proteomics, which focus on the study of proteins, including the structure, function and interactions. So this proteomics involves prediction of involves protein structure prediction, identification of protein-protein interactions, and analysis of post-translational modifications. Then we have structural biology, which focuses on analyzing and predicting the three-dimensional structures of biological molecules. Then we have functional genomics, which is for understanding the functions of genes and their interactions within the genome. So others include phylogenetics, systems biology, metagenomics, pharmacogenomics, transcriptomics and bioinformatics. So now I have one more question. So in which process of the central dogma does the synthesis of functional proteins occur? Yeah, so the answer is translation. Then what is the primary focus of genomics in bioinformatics? Whether genomics focuses on studying protein structures or analyzing evolutionary relationships or exploring the entire set of genes within an organism or predicting the three-dimensional structures. I'll wait a minute so that others can answer. So the question is, what is the primary focus of genomics in bioinformatics? Whether genomics focuses on studying protein structures or whether genomics analyzes the evolutionary relationships or genomics helps in exploring the entire set of genes within an organism or genomics helps in predicting three-dimensional structures. Yeah. So one more information here. All these answers or This questions in problem solving session are only for practice purpose. These are not graded, so everyone can feel free to give their answers whether it is right or wrong, as these answers are not recorded or graded. Yeah. So genomics deals with the exploring the entire set of genes within an organism. Then we'll start with the sequence analysis part. So sequence analysis in bioinformatics is a fundamental and crucial aspect that involves the examination and interpretation of biological sequences such as DNA, RNA and protein sequences. These sequences carry the genetic information essential for understanding various biological processes. So DNA sequence means specific order of nucleotide bases, that is adenine, thymine, cytosine and guanine. So the DNA sequence serves as a fundamental code that carries genetic instructions for development, functioning and maintenance of living organisms. Each DNA sequence is unique to an individual and is inherited from one generation to the other. So the DNA structure is composed of 4 nucleotide bases, that is adenine, thymine, cytosine and guanine. The sequences of these bases along the DNA molecule is what DNA sequence means, and in the DNA sequence we have complementary base pairing, that is A pairs with T and C pairs with G. So this complementary base pairing ensures the stability of DNA double helical structure. The sequences of base pairing is crucial in understanding the genetic code and relationships between different DNA molecules. Bioinformatics tools are used to extract meaningful information from DNA sequences, which aims to understand the functions of genes and non-coding elements within the genome. Then we have proteins, which are essential macromolecules in living organisms performing diverse functions such as enzymatic catalysis, structural support, signaling, transport. So protein sequence information refers to the specific order of amino acids in a protein molecule. The sequence of amino acids in a protein determines its structure, function and interactions with the other molecules. Proteins are composed of 20 different amino acids, each with unique chemical properties, that is, each differs in their side chain, that is the R group. Each amino acid is differed by the presence of different R group. So the sequence of amino acids in a protein is dictated by the sequence of nucleotide bases in the corresponding gene. The primary structure of a protein refers to the linear sequence of amino acids and the order of the primary structure of a protein refers to the linear sequence of amino acids, and the order of amino acids is determined by the genetic code encoded in the DNA of a gene. Amino acids are linked together by peptide bonds to form a polypeptide chain, and the sequence of peptide bonds determines the primary structure of a protein. So sequence of nucleotides refers to the DNA sequence and sequence of amino acids refers to the protein sequence. So sequence alignment, motif identification and structural prediction are the common bioinformatics approach that are applied to protein sequences. So now I have a question, like how many different types of amino acids are present in proteins considering naturally occurring? we have 20 different amino acids, and the order of amino acids is determined by the genetic code encoded in the DNA of the gene. Then here in DNA, what is the complementary base pairing that ensures stability of double helix structures? Yeah, as I mentioned before, these sessions are only for practice session. The answers are not recorded and evaluated, and most of these problem live solving sessions will have questions that are similar to your assignment questions. Yeah, so here do check for multiple options which may be correct.
Sir, option B and D.
Yeah, yes. Yeah, so here both option B and D are correct. So A pairs with T forming 2 hydrogen bonds, and C pairs with G having 3 hydrogen bonds between them. Yeah, okay. There is one question in the chat box. This type of model question can be get in the final exam. So this is a concept related question that I have made. Further, I'll also see how you may get numericals in exam using this concept of complementary base pairing. I have created some of them in the next coming slides as well. So next is sequence analysis. So decoding messages is also key step in gene expression, in which information from a gene is read out to build a protein. So codons. So cells decode RNA by reading their nucleotides in groups of 3 called codons. So the codon table specifies the correspondence between nucleotide triplets. That is, nucleotide triplets are called as codons. So the correspondence between nucleotide triplets in mRNA and amino acids they encode during the process of translation. So this codon table it serves as an universal guide for translation of genetic information into proteins, ensuring consistency in protein synthesis across different organisms. So there is one concept called genetic code redundancy. The genetic code is degenerate, meaning that multiple codons can code for the same amino acids. The codon table here shows the redundancy in genetic code, allowing the flexibility in DNA sequences without altering their protein sequences. So this redundancy is also known as wobble hypothesis, where the third position in codon is less stringent, allowing for a variation. Followed by this regular 20 amino acids coding, we also have start and stop signals. The codon table designates specific codons as start and stop signals. Yes, AUG serves as the universal start codon, and it also codes for methionine initiating the protein synthesis. Then we have UAA, UAG and UGA, which are stop codons signaling the termination of protein synthesis. So the table here provides a comprehensive list of codons and their corresponding amino acids. So this information is crucial for understanding the primary structure of a protein. So, and there is one question in the Google forum as well. The question is regarding whether one has to remember the codon table. So it may not be mandatory to remember the codon table, but at least to remember what is a start and stop codon and number of codons that are specifically coding like number of codons each amino acid has. So this basic information would be sufficient. Yeah. So there is one more question in the chat box, but still, yeah. C pairs with G provides more stability because it binds with 3 hydrogen bonds. Yeah, yes, that is correct. So the melting temperature or the strongness or stability of a DNA is calculated based on ATGC percentage also. So that calculation of 80 percentage and GC percentage is also will be discussed in the upcoming slides. So a reading frame in a DNA or RNA molecule, a reading frame refers to a specific way of grouping nucleotides into sets of 3 known as codons during the process of translation. Translation is the conversion of genetic codon carried by mRNA into sequence. The reading frame determines the order in which these codons are read, and each reading frame may produce different sequence of amino acids. So here I have given 3 possible reading frames for 5' 5' to 3' of a mRNA. So can anyone mention what is the difference between these 3 reading frames? What is the change we are observing? In what way frame 1, frame 2 and frame 3 is different from others? Other than stop codons, any other? Can anyone observe the start position?
Sir, I think there are different frame of references as considered as the first three cases.
Yeah,
So the first one is started from U, and second one is starting from CAU, and the third one is AUG.
yes, yeah, that's correct. Thank you. So these 3 reading frames are established by selecting a. Someone has raised a hand.
Yes, sir. Sir, I think the difference between these three is that the first frame and the second frame would not really produce a protein because AUG is not the first codon.
Yeah, that is
The
also.
proteins would be truncated.
Yeah, yes. Thank you for reminding. Yeah, that is also one case. So here I wanted to tell that 3 reading frames are established by selecting a starting point. So first starting frame starts from the first amino acid, first nucleotide. Second reading frame starts from the second, and third reading frame starts from the third. So in first one starts with U, second one starts with C, and third one starts with A. So in this way, if you segment these nucleotides into codons, the codons what you observe in 3 frames are different because of one positional shift. So here I have a question, why not frame 4 is available for this? Can anyone think of an answer? Why there is no frame 4, frame 3 is last?
Because
Sir,
that will be similar to frame 1.
stop codon UGA is a stop codon.
Yeah, I think the first answer is correct, because if you consider frame 4, which has to start from UGA, so already the part of protein that starts with UGA is present in frame 1. So since we have triplet codons, triplet nucleotides forming codons, so we have 3 frames for 5' to 3', then we'll have 3 more frames from 3' to 5'. So for example, here consider the DNA sequence. Now I wanted to convert this into 3 possible reading frames. So the first reading frame starting from first nucleotide. So ATG is one triplet codon, a triplet nucleotide, one codon. TCG is one, GAA and TAA. Then frame 2, which starts from second nucleotide, that means it avoids A and start with TGT. Similarly, we have frame 3, which is starting at the third nucleotide. So, yeah, here I have few questions that are out of context but could be useful to understand how a codon table is formed. And so try to answer for the first one now, that is how many two letter words can be formed with letters A, B, C, D allowing repeats. Yeah, okay, let's move on to my workbench. So we have four letters to get filled in two positions. So number of ways we can fill this position is 4, and number of ways we can fill this position is also 4 ways. So the total number of ways will be multiplication, as filling of first position and second position is independent of each other, so we have 16 ways. So similarly, now try to solve the second one. So second question, I'll give some time. So the first answer is 16. So how many 3-digit numbers can be formed with digits 1, 2, 3 allowing repeats? Yeah, so regarding this problem solving session, in the announcement, the time of the problem solving session will be posted along with the access through the recorded video session, the presentation slides, as well as any other extra material that is required. So now that given we have 3 digits to fill in 3 places, so we have 3 places to get filled. So first place,
என்னடி வச்சிருக்கீங்க அவங்க க்ளீன் பண்ணுறது
yeah, so here we have 3 places. So first place we can either fill 1, 2 or 3. Similarly, in second place also we can fill 1, 2, 3. Third also C. So 3 into 3 into 3, that is 27. So given this, can someone mention how many possible codons, in how many ways a codons can be formed, considering a given DNA or RNA sequence? So yeah, 64 is correct. Similarly, codon is a triplet nucleotide, so it has 3 positions, and in codon we also allow repeats. So first place we can fill with either A, D, G, C in terms of DNA. Similarly, in second place also we can fill by 4, third place also 4. So it gives 4 into 4 into 4, that is 64. Yes, there are 3 stop codons and 1 start codon, which codes for methionine, followed by restol. So rest 60 codons code for 20 amino acids, because multiple codons code for similar amino acids. So this is called codon degeneracy. So let's start. So we have then, yeah, how many codons code for a stop codon is genetic code? I think the answer is already in the chat box. Yeah, so the video recording as well as the slides what we are teaching throughout the session will be available along with the clear solutions also, but it may take 2 or 3 days to get posted. Yeah, thank you. Okay, let's move ahead. So we have 3 codons, code for the stop codon. So the first step in sequence analysis I want to start with is nucleotide composition. So nucleotide composition refers to the distribution of ATGC nucleotides in a DNA or RNA sequence. So understanding the nucleotide composition is essential for analyzing and interpreting genetic information. It provides insights into the genetic code, regulatory regions, and structural characteristics of DNA or RNA molecules. So first we have AT and GC content, that is percentage of A and T in a DNA sequence and percentage of G and C. So AT content represents total number or a percentage of adenine and thymine nucleotides in a DNA molecule. So it is a measure of purine and pyrimidine balance in a DNA sequence. Then we have GC content, which represents the total number or percentage of guanine and cytosine nucleotides in a DNA molecule. It is a measure of hydrogen bonding strength between complementary base pairs. Followed by that we have amino acid composition. Amino acid composition refers to the types and proportions of amino acids present in a protein molecule. So proteins are composed of amino acids linked together by peptide bond, so that there are 20 different amino acids, each characterized by a specific side chain. So understanding the amino acid composition is crucial for understanding structure and function of a protein. It influences the protein properties such as folding and interaction with the other molecules. So changes in amino acid composition can impact the biological activity of a protein. So given this nucleotide composition, amino acid composition, AT and GC content, I have made some questions. The first question is, if AT content of a DNA sequence is 28, what is the expected percentage of GC in the same DNA sequence? Yeah, let's move on to notepad again. So given AT content equals to 28%, so we can also consider A equals to T, so which will be 28 by 2, so it is 14% of each. And we also know DNA is made up of completely ATGC only, so which forms for 100%. So GC content will be 100 minus 28, which equals to 72. So moving forward, I have one more similar question. It is 72. So these are according to Chagraph rule. So the amount of nitrogenous bases were not found in equal quantities. So this rule states that DNA of any species or any organism, the amount of guanine should be equal to the amount of cytosine, or the amount of adenine should be equal to the amount of thymine. So try to solve this question, where if the T content in a DNA sequence is 12, what is the expected percentage of GC in the same DNA sequence? I'll wait for a minute for others to solve. Okay, so according to the Chagraph rule, we know A content should be equal to T, so which both will be equals to 12. So AT content will be 12 plus 12, that equals to 24. Then we know AT plus GC content should make up to 100%. So therefore GC percent will be 100 minus 24, that is 76. So given G content will be 76 divided by 2 and C content will be 76 divided by 2 is 38. So I hope everybody is clear how this percentage calculations has been made. If anyone wants to know again, I can explain them. Do let me know. So after this I will be starting discussion about some of the biological databases. So mostly I will be discussing about the sequence, pathway, disease and other biological So in bioinformatics, a wide range of databases is available to store and manage biological data. These databases cover diverse aspects of biological information including genetic sequences, protein structures, functional annotations and more. So here are some of the examples of biological databases. So biological structure databases include PDB, which contains the three-dimensional structure of biological molecules, primarily proteins and nucleotides, and this PDB database is essential for structural biology, drug discovery and understanding the molecular interactions. Then we have a nucleic acid database, so which mainly contains information on experimentally determined nucleic acid structures including DNA, RNA. So this supports the research in structural genomics, RNA biology and DNA protein interactions. Followed by that we have some more structure databases that are hosted by Europe Bioinformatics Institute. Then we have protein sequence database. So under protein sequence database we have two main categories, that is protein sequence database and nucleic acid sequence database. So GenBank is a comprehensive database of nucleotide sequences including genomic DNA, mRNA and protein coding sequences. And then we have UniProt, which is a comprehensive resource for protein sequences and functional information including entries for manually curated and computationally predicted data. So this UniProt is essential for protein research, functional genomics and proteomics. Followed by that we have other databases which are related to functional and pathway. So we have KEGG, which provides information on biological pathways, diseases and functional annotations of genes and proteins. So this understanding of these biological pathways, disease and functional annotation facilitates the understanding of cellular processes, pathway analysis and systems biology. Then we have clinical and disease related databases as well, such as ClinVar. So this ClinVar database will have information about clinically relevant genetic variations and their associations with the human health or diseases. So this database helps to understand the interpretation of genetic variants in the context of clinical relevance and diseases. Then we have TCGA, that is the Cancer Genome Atlas, which has genomic and clinical data related to various cancers. So now, so these are the questions that are mostly relevant or in par with your assignment. Just please, someone has raised the hand.
Sir, in the previous slide, there is the mentioned primary, secondary, and derived databases. So, what are these? Please, can you tell us?
Yeah, so primary databases uh, are the first source of the database, and derived things databases are where databases collection from the other databases. So secondary databases will have the hyperlinks to the other data. For example, if I am mentioning a protein stability information in my secondary database, I create hyperlinks to my primary database. Primary database here is PDB, from where the main source information is collected, followed by some annotation work is done. The exact created database is primary, and collection of databases can be given as a derived databases. Secondary is the collection followed by some manual intervention or manual annotation or analysis is secondary databases. So we have a quiz question starting, which is average hydrogen bond energy for the sequence ATAGC. So given the hydrogen bonding energies are given in brackets. Yeah. So here the hint is try to consider the overlapping nucleotide segments from a given sequence, then try to find the sum of those values divided by the number of overlapping segments that you have observed. So why one has to consider overlapping segments is because they provide more accurate representation of interaction, particularly in structures and in terms of thermodynamic properties of these nucleotides. So this considering overlapping nucleotide segments lead to more precise energy estimations in computational studies. Okay, first try to identify the overlapping dinucleotide segments. Let's move on to workbench. So this is the given nucleotide, so I'll make possible overlapping dinucleotide peptides, nucleotides. So given a sequence of length n, so number of dinucleotide peptides will be n minus 1. So observe sequence length of 5, so we have 4 dinucleotide peptides. Then take the values for each of them. The average hydrogen bonding energy for AT is -7, TA is also -7, AG -9 and 12. So sum up these. This sum will be coming up to 35. So final value will be 35 divided by 4, which is approximately 8.7. So first initially I have considered all possible dinucleotides, then calculated sum of their average hydrogen bonding energies, then sum divided by number of dinucleotide peptides. So now that I have given number of dinucleotide peptides for a given sequence of length n is n minus 1, what will be the number of trinucleotide overlapping trinucleotide tides you can see for a sequence of length n? Given number of dinucleotides you can observe for a given sequence length n is n minus 1. So number of trinucleotides we observe is exactly we observe n minus 2. So here the answer is minus 8.75. So now number of overlapping trinucleotide units obtained for a DNA sequence length of 20 nucleotides is okay. Let's understand this a little more sequentially. Consider a nucleotide sequence of length 6, and now let's try to form the possible trinucleotide units the two overlapping trinucleotide units. We have totally 4, so the formula is n minus 2, where n corresponds to the number of sequences, length of the sequence given. So in my question the length of the sequence is 20, so 20 minus 2 equals to 18. This is the answer. Yeah, next question is, given a DNA sequence CG, TTT, GSE, how many hydrogen bonds would be there between base pairs? Okay, I'll wait for some more time for others to answer. So the answer I see is 20. So this is the sequence given. So C can form 3 hydrogen bonds, G can form 3 hydrogen bonds, T can form 2 hydrogen bonds, T again can 2 hydrogen bonds, 2 hydrogen bonds, 3 hydrogen bonds by G, 2 and 3. So I'll try to fill out the sequence from back. So C has complementary with G, D, C, A, so let's count the number of hydrogen bonds here. 3 plus 3. So given the sequence, the number of hydrogen bonds it could form through base pairing is 20, and this is the complementary stand for the given sequence. So if we consider this is 5 prime to 3 prime, we can consider this as 3 prime to 5 prime. Further, I have some more questions regarding the complement Right, base pairing has solved. So given a DNA sequence from 3' to 5', what will be the complementary stand for it? So remember, the complementary base pairing is A pairs with T and G pairs with C, and try to answer this question. Yeah, I see some answers coming up in the chat box. Everybody have been voted for B. This is the given nucleotide sequence. So A pairs with T, T pairs with A, G with C, C, G, A, C, T. So 3' becomes 5', 5' becomes 3'. So this is the complementary strand for the given sequence. Here the answer is B. Yeah. So how many nucleotides are needed to code for a protein that is 100 amino acids long? So here the question is, how many nucleotides are needed to code for a protein that is 100 amino acids long? Either 100 nucleotides code for 100 amino acids, or 300 proteins code for 100 amino acids, 1000 code for 100 amino acids, or 3000 code for 100 amino acids. Yes. So we know triplet nucleotides which form a codons. So 300 nucleotides will code for 100 amino acids. So now I have a true or false based question. Is it easy to break the bond between A and T than in between G and C? Whether it is true or false? So why it is easy to break A and T? Why not G and C? Okay, understand answer is true. Yes. G and C, a bond has been formed by 3 hydrogen bonds, and A and T has 2, so it is easy to break A and T than G and C. So which among the following in Emboss is used for finding the complementary stand for a sequence? So reverse-seq is used here. Trans-seq is used to translate the protein. Back translate is protein to nucleotide, and 6 back is the one which gives the 6 reading frames of a protein, a nucleotide sequence. So DDBJ is one of the famous database, and it's also part of the international bioinformatics collaboration. So what does DDBJ stands for? Okay, everybody tells DNA is DDBJ is DNA data bank of Japan. So which of the following has only a single codon? For this question you can refer to the codon table online. Yeah, so tryptophan has only one codon that codes for itself. Followed by that we also have methionine, which is a start codon. It is also coded by single codon. So I mentioned in the discussion during data banks. So GenBank contains the information on so GenBank is a primary database that contains information on nucleic acid sequences. So here I have a quiz which is for some numbers, numerical based questions. So most of these questions I have discussed during the course lecture course today as well. So try to answer them. So when you are answering, use the Question number like A, what is the answer? B, answer. C, answer. D. I'll wait for a minute more. Yeah. So still answers. So how many different types of nucleotides are found in RNA? So we have 4: AUGC, how many different types of amino acids are present in proteins? These are 20. How many amino acids are needed to code for single amino acid? That is 3. Triplet, we call it a codon. How many different codons are there in genetic codon? 4 power 3, that is 64 number of different codons. How many code for amino acids? Is 61. The remaining 3 are stop codons. Now I'll start with a demo to emboss. So the installation part and all, I'll be uploading a document along with this. So today we'll be using this online website for emboss. So this is the emboss explorer. Yeah, this is the home page. I hope everybody can see my emboss page. Yes. Can anyone reply that my emboss page is visible? Yeah, okay. Yeah, okay. Thank you. So first I wanted to start with the one that is used for complementarity. So as you guys mentioned, it is used for reverse complementarity. So we have RevSeq here. You can search for RevSeq. So I can click on RevSeq. So under the title you can see a brief description: reverse and complement of a nucleotide sequence. For the further information, one can also access the manual of this. So this gives what is the input you have to give and what are the different flags it has. Okay, so I wanted to see what will be the reverse complementarity of ATAGCT, and it is asking us, I'll zoom a little more. I have given us some random sequence as an input here, and it is asking whether we want to print a reverse complement of a sequence or a complement of a sequence. So I am selecting yes. Complement sequence is also yes. The output is FASTA format or any other format is fine. So I am clicking on run revseq. So this is the complementary sequence of our given strand. So let it be the reverse complementarity. So further I want to explain how translation happens here. So similarly I want to search for six pack. So displays a DNA sequence with 6 frame translation and ORFs. So given some random sequence here. So further several options has been used, code to use. So this standard code is better. If there is some organism specific codes as well, so which understands their own organism based translational recognition elements. Followed by that we have other options to mention ORF at the beginning of the sequence, ORF at the end of the sequence, and to start with M or no, that is meter start codon or no. And display translation at reverse sense. This means this will show all the possible 6 reading frames, and one can also mention the minimum size of ORFs that is required and regions to mark in color or to observe. So I am not changing any of these default parameters and click run. So the 6 frame translation proteins has been given. So frame 1 from forward sequence, frame 2 also, frame 3, then we have 4, 5, 6 frames from complementary sequence of the given DNA sequence. Other than this, we have also n number of features, that is extract sequence features, and we also have back transseq, so which translates a protein to its ORFs or a gene sequence. So who is someone completely from biological background or has no background of mathematics or coding, so these types of Emboss Explorer are easy and handy in terms of learning a bioinformatics related concepts. So I have given some practical questions, like it's like take home messages for you. Followed by that, I have a quiz which is using emboss. So for sequence analysis more, we have our one more in-house tool, that is seek2 feature. So seek2 feature is a web server which is a comprehensive based with feature extraction tool which computes protein and DNA sequence driven features. It can calculate nearly 252 protein based and 42 DNA based descriptors. So the major protein based descriptors include physiochemical, energetic and conformational properties, mutational matrices and contact potentials. So when you click on compute here, it asks for either we want to do a sequence analysis of a protein or a DNA. So let me select a DNA, and it also gives whether you want to know information about effect of mutation on DNA properties or average property. So average property is the property value of each nucleotide divided by number of nucleotides. So under this DNA properties, we have physiochemical properties, conformational properties and nucleotide content. So let me select the average property. So since it's asking in FASTA format starts with a header followed by sequence. So it asks for what type of properties you need, then one can click submit. So first we see a list of physiochemical properties along with their scale unit and their average value. So for the given input sequence, the stacking energy, average stacking energy is 1.62 kcal per mole. Further, we also have conformational properties such as bend inclination, major groove depth. So the more information of these properties and explanations will be provided in discussion week on nucleotide. It's either second week or third week. Followed by that, we have also AT content, adenine content, GC content, cytosine content, other. So we have also provided option to download the result. So one can download the results and save for their further analysis. So this is about seek2 feature tool. So using this practical sequence, I will show you how to access emboss and seek2. So I am copying the sequence. So reverse seek, so which is used for finding the complementary strand. So paste your sequence. Since it's asked for reverse sequence only, I am keeping the option as yes. Run. So this is the reverse complementarity sequence for our given input sequence. Then I wanted to find the protein sequences. Similarly, I am pasting the sequence here. The sequence is very large. Emboss also has a file upload option. So I am keeping all the default parameters as such. Then run 6pack. So number of ORFs has been given, and there is subpart question for this. The part is identify the reading frame which this particular protein sequence corresponds to. So let us identify. Say fourth frame corresponds to the protein sequence that is given, so which is of 52 amino acids. So let's try something with the seq2 feature as well. So seq2 features strictly accepts only FASTA format. If anyone doesn't follow this, there is an error displayed, that is kindly upload or either enter the valid input sequence. So make sure that for seq2 feature it is only the FASTA format. So it accepts only the FASTA format of the input sequence either for nucleotide or a DNA sequences. So these are, have created some practical and quiz related questions which I'll provide you as a self practice test because it is already more than 1.5 hours. And also have some database searching methods. So try to find how many Homo sapien sequences deposited in DDBJ and compare with the EMBL. Yeah, so that's all for today. Now I'll take any questions from you. So if you have any other questions, please let me know through unmuting yourself or through chat box.
Hello, good afternoon, sir. Sir,
Afternoon.
good evening. Sir, is everyday class will be start from 6 p.m.?
Yeah, actually this is only a weekly problem solving session. So my session will be every Monday 6 to 8.
Or every Monday 6 to 8. And another other day classes, I
Yeah,
actually I joined a little bit later in this program. So I'm just watching the videos uploaded
yeah.
in the
So
session
in
in
my
blog.
knowledge, there are other two TAs also taking the sessions. One
Okay.
takes on Friday, another takes on Saturday. But exact timings you can find in the announcement section of the NPTEL Swayam portal.
Okay. Thank you.
Yeah, so yeah, my session will be weekly once, Monday 6 to 8 for 12 sessions I'll be having. And further in other sessions as well, once the deadline for assignment gets over, I'll be also discussing solutions for the assignment problems. And so whatever the problem solving sessions, the attendance, the answers are not recorded as well as are not graded. So this is just for a practice purpose. Only the assignments that you submit in the NPTEL portal are graded. Yeah, all the recordings
Sir,
along with the, yes please.
sir, attendance in the problem solving assessment is also recorded, or it is mandatory
So this
or
attendance
not?
is not uh, required, but this is, we feel like a live session is more good for a one-on-one doubt solving. So if you are having doubts, so we encourage you to attend these live problem solving sessions. So this
Okay,
is
sir.
my third year working as a TA for NPTEL, similar course. So live sessions would be more comfortable for a problem or doubt solving. And also recordings will be available for all the live sessions. So recording may take 1 day to process. So by tomorrow night or Wednesday morning, the recording will be available to you along with the material solutions. A drive link will be provided to you. Follow, and also YouTube based recording will be available. Further, if you have any doubts in the YouTube lecture video, you can also comment on that YouTube lecture video. I'll be answering those. I'll also provide my mail ID in the chat box, so that will be easy.
Sir, where we will be getting the links for the materials?
Yeah, in the announcement section of the Swayam portal.
Okay.
So, any other questions regarding? Yeah, even the joining links for the other sessions and my session, every week a notification mail would be posted on the announcement portal on the NPTEL, and when the week lectures are available, the lectures record, the principal investigator or Michael sir's lectures will also be posted. Information about those lectures will also be posted in the NPTEL Swayam announcement tab. Yeah, for those who are joined late, I'm repeating again. So these live solving sessions are not graded. This is only for the practice purpose and doubt solving sessions. So it's a one-on-one training sessions one can expect. Yeah, so I feel that's all for today. Thank you everyone for joining.
Thank you, sir.
So I'm ending the session now.